Okay, guys, today's lecture is all about traits and probability. And so, specifically, we're going to get into a non-Mendelian genetics. And so, what exactly does that mean? Well, we'll talk about that in a minute. So, what we've been talking about thus far is really Mendelian genetics. So, that typical 3-to-1 ratio and that with that F1 cross. So, going from two purebreds, letting them self-pollinate, and then arriving at what you see here on the screen right now, which is a heterozygote for each, both the mom and the dad. So here's mom plant, dad plant. Notice they're heterozygotes, and they're heterozygotes because they have one capital letter and one lowercase letter, which means one dominant gene, and that's this one, and one recessive gene, and that's that one. And dad has the same thing. All right, and so what do you get in the resulting cross? Well, basically you get a 3-to-1 ratio of the dominant, right? So this guy's going to be dominant, this guy's going to be dominant, and this guy's going to be dominant. The reason for that is that recognize that they all have at least one copy of the T. And so because they have one copy of that T, excuse me, because they have one copy of that T, they actually express phenotypically that dominant phenotype. Now, if we look at the one, or in this case, the 3-to-1, the lowercase t, that's two copies of the recessive gene, which is the only time where phenotypically in normal Mendelian genetics you would see whatever that difference is. And so in this case, it's going to be the shorter plant. Okay, and so this is a 3-to-1 ratio. But also, if you look at the genotypic ratio, it's actually 2-to-1, right? Because we have two heterozygotes, one homozygote dominant, and then one homozygote recessive. Okay, so this is normal Mendelian genetics. And so just a reminder as to what we were talking about last class, which is the difference between genotype and phenotype. So the genotype is what is expressed in your DNA. What chromosomal material do you have? It doesn't necessarily indicate what your outward phenotype is, right? Your phenotype is the physical appearance that you have on the outside. So in the case of the plant, is the plant tall or short? It's short. But it does have the genetics, in the case of the heterozygote, it does have the genetics, this tall plant here has the genetics to be short as well, even though it happens to be tall. So genotype versus phenotype. And so now, when we looked at Mendel's P's, remember that F1 generation, those hybrids all looked like the dominant homozygous parent. And the reason for that is because they were all heterozygous. Okay, so remember, this would be the case where we crossed a purebred homozygote with a purebred recessive. These guys would all be this. And then this generation would represent what we saw in the previous slide. And so what would happen if we didn't actually, if this dominant gene didn't kind of overcome the expression of the recessive gene? So what would happen if it didn't have essentially enough power to fully express dominance over the recessive gene? Well, in that case, what you would have is incomplete dominance. And so where exactly would you see this difference? And do you think it's either in the genotype or the phenotype? And since I have it written down right here, so basically what you would see in incomplete dominance is that you wouldn't get either yes, that's what I wanted. you wouldn't get either the dominant or the recessive phenotype. What you would actually get is more of this kind of blending of the two. All right, and so in the case of these chickens, for example, what you end up getting is what they call the blue chickens. All right, and so let's write that out so that you guys can understand how we arrived at that. All right, and so basically what happens when you cross a purebred black chicken with a purebred white chicken. And so when we talk about color, and color is a thing that's blended a lot in incomplete dominance, when you represent color, what we like to do as scientists is we like to represent that as C's. Okay, so if my color is pure black, all right, then that's what the genotype is. If my color is pure white, then that's what the genotype is. Okay. Now what happens when you cross those? So let's do pure black on the top, pure white on the bottom. You would get this guy becomes BW, BW, BW. Okay? And so these guys all become blue chickens. All right? Now, what would happen if you allowed those blue chickens to then mate with one another? So this would be just like when we talked about Mendelian genetics, this would be self-pollination, all right? So now we're just saying we're selecting just blue chickens to breed with one another. Let's represent that now. And so if they're all blue, then they're all basically CBCWs. And so in this case, you get pure blue, then you get, you get pure black, and then you get blue, you get another blue, and then you get pure white. All right, and so this gives you the 1 to 2 to 1 genotype, and also in this case, also the phenotype. Okay, so remember back to Mendelian genetics, we talked about you would only get this in the genotype, you wouldn't actually get it in the phenotype, the 1 to 2 to 1 ratio. With incomplete dominance in this F2 generation, you always get it in the genotype and the phenotype, because remember, now your dominant gene isn't completely dominant over the recessive gene, and because of that, you get the mixing or the blending, therefore giving you a third phenotype. So that's incomplete dominance. Now let's talk about codominance. And so the most common type of codominance that you'll encounter in genetics is going to be blood typing. All right. And so in blood type, basically what happens is both the alleles are simultaneously expressed. So it isn't like there's this mixing or blending. They are both present and both expressed. And so in humans, we basically have 3 different alleles that represent blood type. All right, so and they're designated by the letters A and B. All right, and so either you have this particular carbohydrate chain or you don't. So if you have it, you're A or B. If you don't, you're represented as an I. And so if you are, for example, II, as it says here, you are actually type O. So type O blood is actually blood that doesn't have the carbohydrate chains for either A or B type blood. All right, and so if we look at these, how many possibilities do we have in terms of blood type? Well, we have type O, which is no carbohydrate chain. We have type A, which can either be homozygous for A, or you can be heterozygous. You can be A on one of those alleles, but on the other you could have no representation of a carbohydrate chain, which would make, essentially that would be a parent that was type O blood. And then same thing here for B, homozygous for B, and basically a parent that gave you O blood. And then you could also have a parent provide or give you type AB blood, and that would be from an AB parent or a type B or A parent. And so there's 4 types of blood types, and these are the genotypes for them. Okay, so the phenotype will be the blood type here. The genotype will be the actual genetic information that they have. Okay, so what happens when you have essentially more than one set of alleles responsible for a particular phenotype? All right, so in part, skin color is represented this way. So there's actually multiple alleles, multiple genes that actually code for a skin type. But going back to blood, there's actually 2 parts of blood. So if you guys know your blood types, you're going to be A, B, AB, or O, but then there's this other component of your blood type, which is very important, called the rhesus factor. And so that determines whether you're positive or negative. So when you say you're AB positive, it means that you have the carbohydrate chains for A and B type blood. But in addition to that, you also have the D antigen, which is the rhesus antigen. And that's Rh positive, okay? We represent rhesus factor by basically capital D and lowercase d. It just makes it easier than writing down all the RHs. So Rh positive is going to be either DD, capital D, lowercase d, or capital D, capital D. The only way you're Rh negative is if you're lowercase d, lowercase d. All right? Okay, so let's actually do a dihybrid cross for blood type. And so a dihybrid cross is when you're looking at more than one set of alleles. Okay, so in this case for blood type, we're looking at not only the alleles for whether or not they have the carbohydrate represented, so meaning are you A, B, AB, or O type blood, but also then whether you're Rh positive or Rh negative. Okay, and so in this problem that's set up here, it says, what are the phenotypes of parents with respect to blood type? AO is expressed as type A. Rh positive, Rh negative is actually expressed as Rh positive, and so the father's phenotype is A positive. The mother is BO, and she's expressed as type B, and her Rh factor is positive, and her blood type is B positive. Okay, and so if we look at this, how do we represent this? And so on the next slide, they're going to represent it writing down the Rh factors, but I want to show you guys how it's a little bit easier to track the different genotypes. You can represent blood type as, so if they're A positive, you can represent it as AO. You can also represent it as such. So it can be IAI. All right, and then this one would be IBI. Okay, and the reason why I put that is because notice that both these guys have the O. And so basically they have an empty carbohydrate chain, which is this guy here. All right. Oops. Okay. So what then are the possible genotypes for their offspring with respect to their blood type? Well, so what you can see here is the dihybrid cross. And so one thing that you have to do before jumping into the dihybrid cross is you need to figure out what are the possible gametes going into it. And so it's nice and easy when you have just a single allele that you're representing. But when you have 2 alleles that are being represented, basically you have to figure out what are all the possible contributions from those parents' gametes. And so if we look at the father, all right, so remember we said dad was IAI, and he was Rh positive. but he did carry that negative phenotype. And so we have to figure out then, what are all the possible combinations? And so this is where basically we go ahead and we start crossing them. All right, so in order to figure out the gametes, remember you can only have one of each of the alleles. All right, and so for the first cross we have IA capital A, excuse me, and that will cross with RH positive, and then this guy with negative. All right, so one possible representation is as such. Okay, and then if we go on the bottom, when we go to the other allele, we have little I, little I. Okay, and so these are the 4 different gamete possibilities from just the paternal gametes. All right, so 1, 2, 3, 4. Okay, now let's write mom on the left-hand side here, and let's figure out her possible gametes. So mom is BO, all right, which means she's actually IBI, and she's RH positive, but she has the information for that rhesus negative factor. She's heterozygous. Okay, so the cross here, possible gamete number 1 is going to be IBRH positive. Possible gamete number 2 is going to be IB IBRH negative. And then gamete number 3 and 4 is going to be IRH positive, IRH negative. Okay, so recognize this. So 1, 2, 3, 4 gametes here as well, and those are represented here on the y-axis, and the dads are represented here on the x-axis. And then basically you just cross them just like you would. So here you can see type A and B are represented, and then you get RH positive and RH positive, which gives you AB, RH positive, RH positive. Okay. So that is your first dihybrid cross, and that is representing multiple alleles, multiple chromosomes there. Okay, so here's another dihybrid cross, and this time instead of looking at blood type, you're just looking at 2 different traits. So the first trait is the color, and then the second trait is going to be the shape of the pea, so whether or not it's round or kind of the wrinkled. Okay, so if you just look at this, there's a lot of information that this first picture gives you here at the top. So this guy, basically what it means is that in order to be green, you must be homozygous recessive, and in order to be wrinkled, you must be homozygous recessive as well. Okay, which then means that the dominant gene is yellow, and the recessive gene and the dominant gene in terms of the shape is round. So just that first picture tells you all of that about that parent generation, okay? Because in both cases you have the purebred form of both the color and the shape. Notice in the F1 generation, just like we've always gotten before, basically you get a 50-50 split of the gene pool, so you end up having heterozygotes for both color and shape. But this time, even though the representative color and shape are the same as you would have seen in the parental generation, we know that this particular P contains the information for both the recessive traits. So if you were to allow then this particular P to cross itself, okay, to self-pollinate like we've talked about before, what would happen? All right, and so basically you have to do the same thing we did before. with the blood typing, except this time it's a little bit easier. We're not writing down I's or anything like that. We're just writing down the different letters representing each of the traits. Okay, and so if we say, here we have capital Y, capital R, lowercase r. What are all the possible gametes? Well, you just cross them. Okay, so YR, and then YR, and then little YR, and then little r. Okay. And then basically you just do the cross. Okay. Now, before when we talked about that F2 generation with Mendelian genetics, we referred to that F2 generation and that typical monohybrid cross of one gene to be 3-to-1. Here's the special number for the dihybrid cross, looking at 2 traits, and it's 9-to-3 to 3-to-1. Okay, so notice we still kept that 3-to-1, but now we've increased the amount of possibilities because we've increased the number of genes going into it. And so 9-to-3 to 3-to-1 is the representative ratios here for the normal Mendelian dihybrid cross. Okay, so we're going to talk a little bit here about sex chromosomes, specifically in humans, but I did want to mention that not all creatures have the same sex chromosomes. And so if we look at honeybees, for example, they have what we call haplodiploidy. And so basically, the queen bee has the ability to lay either fertilized or unfertilized eggs, and those unfertilized eggs can turn into males and contain half the genetic information and are perfectly fine, except that they are always males. And they can also generate diploid females by basically creating a fertilized egg. And so in this case, the female queen bees actually go around and they collect sperm from many different male bees, and they collect that sperm into an organ that they have for basically sperm collection, and then they can control when their eggs are fertilized and which eggs get fertilized or not. The mammals like us, we are all either XX or XY. So males are always XY, and females are XX. There are a bunch of different genetic disorders that basically cause non-disjunction, and that results in either a single X or XXY. And those are basically abnormal phenotypes like Klinefelter's or Turner syndrome. Birds, basically females are heterozygotes and males are homozygotes. So whereas in humans, females are homozygotes, and in chickens, the males are actually the homozygotes. And then in some crickets and other insects, basically females have the 2 sex chromosomes, an XX, and males basically are missing a sex chromosome, and that's what we call XO. So there are a bunch of different genes that are what we call sex-linked genes. And basically, sex-linked genes are any genes that happen to reside on the X chromosome, on the sex chromosome, excuse me. In humans, I gave it away there a second ago, most are on the X. And if you couldn't figure out why that was, it's basically because if we think about males and females, what do both of the sexes have? They both have the X chromosome. And so if the important genes or the sex-linked genes are located on the X, then we can assure ourselves that nothing goes missing with the males when they drop an X, essentially. It also is helpful for females in that we have 2 X chromosomes, and in having 2 X chromosomes, should something go wrong with the other X chromosome, then we can have what we call compensation. And so one X chromosome could essentially fix what's wrong with the other one. Not fix in terms of do anything about it, but just be there just in case whatever we need to be represented in that X chromosome isn't there. And so sex-linked genes were discovered back in 1910 by Thomas Hunt Morgan. He worked in a fly lab at Columbia. And what he noticed was that white-eyed flies just didn't follow normal Mendelian genetic patterns. And specifically, when he looked at the flies, none of the flies with white eyes were female. And so he thought that was really interesting and just thought, hey, maybe there's something to the differences here between males and females. And so he basically deduced from what he saw that perhaps there were no white-eyed females because the gene is on the X chromosome. And he also noticed that in F1 hybrids, the result from the cross of the red-eyed female and white-eyed male was that all flies had red eyes. Okay, and so if we look here we had the red-eyed female, and then we have the white-eyed male, because remember he only saw white eyes in males. Then when you cross these guys, and so remember we're just cross the sexes, and then each of the sexes, when you have a gene on a chromosome, that is represented by a superscript. And so, for example, so here we have an X chromosome. Whatever we're talking about in terms of the allele is going to be represented by a capital letter or a lowercase letter here. Okay, and so the white-eyed males, they represented as the lowercase r, and the red-eyed, they represented as the dominant gene because the females all had it, but the white-eyed, but the only ones that had the white eyes were the males, and so they represented that as the lowercase r because they assumed that at that time it was probably one of these recessive genes. If we move on to the F2 generation, he noticed that all females had red eyes, but now if we look at half of the males, so this is important because I think in terms of thinking about ratios and percentages, you don't necessarily think about this as half. Okay, so 75% of this population had red eyes, but 50% of the males had white eyes. And the reason for that is 2 of the 4 total offspring from this case are males. 1 of 2 of those males, which is 50%, had white eyes. Okay, so you only had a 50% chance of having a male. Half of those males had white eyes, and so 50% of the males had white eyes. And so the bottom line and what he deduced from this was that basically in order for a female to actually have white eyes, she would need to express 2 recessive genes. So she would need to be XRXR. And males only need the 1 because they're what we call hemizygous. Hemizygous meaning they basically have 1 X and 1 Y. And so because they only have that 1 X, they only have 1 possibility of having any gene represented on that X chromosome. So they only need 1 copy of it to display that outward phenotype. Okay, so now let's move on to human pedigrees. And so this is basically how geneticists, how they go about determining whether or not a particular individual will be affected or susceptible to a particular genetic disease. And so when you look at a pedigree or a family tree here, that's basically, it's just that. It's a family tree, and it records and traces the occurrence of a trait in a particular family. And so there's a couple things you need to know here. Squares represent males. Females are represented by circles. And when you are an affected individual, or when you have whatever that particular genetic trait is, you are represented as a colored in shape. Okay? If you have a non-colored in shape like this is an unaffected female, unaffected male. This is an affected male, and this is an affected female. Okay? And so it's important to know, there's this question here, does this show the individual's phenotype or genotype? it's not showing us their genotype, right? It's just showing us what they have outwardly, what they're showing us. And so that's important because it's not like when you go to get genetic testing, like you've gotten genetic testing on every single generation up into your generation. A lot of times all you have is the genetic information that you're bringing to the table that you've got tested for, but you didn't get genetic information for your mom and dad and your grandparents. And so we can deduce from this what their genetic information might have been, but we don't know for sure what their genetic information is. But like I said, you can actually, if you have enough information on your pedigree here, you can actually determine what the genotype might have been, and kind of backtrack what it might have been. Sometimes there's not enough information, especially when we're talking about homozygous and heterozygous dominant, because those both appear in the same way. So in many ways, it's easier to identify whether or not someone has the recessive gene, because it's only showing if they're homozygous recessive. And then so if we look at the pedigree, in this case, it's just showing whether or not earlobes are attached or not the end of the world. But these are particularly important when we start to talk about deadly or basically disabling traits. And so they're very important when we go to determining whether or not a person's actually going to ultimately reproduce. And so with that, I'm going to end the recording for the day. I will see you in class.